Thank you for the introduction. And thank you for mentioning that my main area of expertise is actually not what I'm going to talk about today. So the main area of expertise is the theoretical aspects of DNA computing. But in the past years, I had some very nice students who dragged me, gradually into this field. So I wanted to mention that, this talk is under the umbrella of the Waterloo AI Institute, but this is not about theoretical advances in machine learning. This is about, how we can improve machine learning with the purpose of specific application in mind. So it's more of an application of machine learning talk, but of course, you have to change your tools depending on the application. So I'm going to talk about the machine learning for the purpose of biodiversity informatics. Today I'm going to try to answer two different questions. I'm going to try to answer the question, does biological kinship produce the detectable mathematical signature in a genome? And the second question, which is the highlight of today's talk, is whether the environment can produce a detectable kinship-independent mathematical signature in genomes. So the first question, does biological relatedness produce a detectable mathematical signature in genomes, falls under the big field of taxonomy. So I'm going to talk about the mathematical representations of DNA sequences. And first, let me define the terms. So what is taxonomy for the purpose of biology? Taxonomy is the branch of science that groups and names living organisms based on relationship inferred by shared characteristics. So the first scientist to attempt taxonomy was Aristotle. He was the first one to attempt to classify organisms. And he subdivided plants into shrubs, herbs, and animals, and shrubs and herbs and animals into air, water, and land. So currently, we don't do that anymore, right? Because obviously, this attempted taxonomy has problems, because this grouping of animals into air, water, and land would group fish, tuna fish with dolphins, right? So there are limitations at grouping animals by the way they look. So then people started to look at morphology, their function, how they reproduce, and to refine this classification system. Currently, the entire taxonomy is based on DNA-based techniques, based on sequencing genomes, looking at the genes, aligning genes, and seeing how genes are related to each other, and that determines the degree of relatedness. So nowadays, we do taxonomy based exclusively on genomic DNA. So what is the general purpose? The general purpose is to take all life on Earth and fit it into one gigantic big tree of life. So here I have two different representations of the tree of life. This is an artistic poster, which I have on my fridge. Actually, I have two copies of it. It starts from here. It has the plants and the animals, and here at the top of it is Darwin. I have a new modern version one that is not sexist and colonialist, and actually it has like several humans of various genders at the top, which I think is more appropriate with the times. I like this one as well. So you see, we want to find out what everyone what everything is related to name, classify, and taxonomically species. So the most important thing that is going to be relevant for the talk of today is that according to the current modern classification system, life is divided into 3 domains, which include 6 kingdoms. So the 3 main domains are bacteria, archaea, these are organisms without nucleus, and organisms with nucleus, eukaryotes, with true nucleus, that have the kingdoms protists, plants, fungi, and animals. So we have basically 3 domains and 6 kingdoms. This is the current view of how life on Earth should be grouped. So we are going to propose some methods that allow us to classify an unknown organism into one of these, place it somewhere on its own branch on the tree of life. Okay, so why is this more relevant today? Well, the study of Earth's biodiversity has become especially relevant today. This is also because of, I was not aware of this before I started to work on this field. I didn't know that 95%, and there is no dot missing there, it's not 9.5, it's 95% of multicellular species on Earth do not yet have a scientific name and are not yet classified. So basically, we know nothing about what exists on this Earth. And I'm not only talking about small things, I'm talking about big things. You know, I also read that every single year since 1948, 18,000 new species per year are discovered each year. And they're not little guys either. So this is from 2020, you know, newly described primate already facing extinction. Okay, there it is. We have a whale that was discovered in 2022 that nobody knew about it before. So there are new organisms discovered all the time. And why this is important is that we cannot protect what we don't know. So if you want to protect biodiversity on Earth, we first have to know that it's there. Otherwise, it could be there and disappear, and we didn't even know we had it. So one of the grand challenges that are part of this big project BioScan that we are part with, that is spearheaded by International Barcode of Life at University of Guelph. It's like a multinational project involving 40 countries, hundreds of scientists. So the biodiversity grand challenge is to map all life on Earth by 2045. And due to current modern sequencing techniques and classification techniques, and hopefully with our help, this is not a, this is not a science fiction project. This is just a science project. Hopefully it's doable by 2045. So this is where our taxonomic questions fall. They fall under this umbrella. We want to find out and map and classify all life on Earth. How do we do that? So what I'm going to use for that is that I'm going to introduce some mathematical representations in genomes. So the traditionally classification of species is based on gene alignment. What do I mean by that? You know, you take a certain gene. For example, you take the gene that is used for the digestion of milk, which is called lactase, you know, the lac operant. You take that gene from various different organisms that have that gene, like, you know, a cow, a human, you know, a sheep, you know, some other cattle. And then you look letter by letter and see how different they are from each other. And you compute this distance. And if the distance is small, they are more related to each other. The distance is big, they are distantly related. So basically, this is how gene alignment works. But our method is not based on aligning genes. Our method is alignment free. So what I'm going to do is that we are going to take a random fragment from a genome and then use some mathematical representation and extract some features that can act as genomic signature. Okay, so what I'm going to talk about first is what mathematical representations of DNA sequences we are using in our methods. First, I'm going to describe what is called the chaos game representation, which is a visual representation of DNA sequences. Then, you know, I'm going to talk about the connection between that and the vector of k-mer counts. What I mean by k-mer is a subword of length k. So if you think of a DNA as a sequence, and let's say that k equals to 6, you have a vector that has the counts of all 6 mers of all subwords of length 6. That's a feature vector for that. That's a numerical representation. And I'm also going to use another representation of a DNA sequence as a digital signal, which would be a numerical vector obtained from, you know, taking a DNA sequence and assigning each letter. I assume that everybody in the audience knows the DNA alphabet. So the DNA sequence is it's a sequence over the 4-letter alphabet ACGT. So the digital signal transforms each of these letters into a number. And this number is either a number without a biological significance or it corresponds to some physiochemical properties of that letter. So these are 3 different mathematical representations that we used in our research. So let's start with the first one, because it was, it was our first and also is the most fun one. So the chaos game representation of a DNA sequence works as follows. We start with a unit square whose corners are labeled A, C, G, and T. And the first pixel of the figure, so I want to produce a visual representation, is the center of the square. Then we read the DNA sequence that we want to plot from left to right. And with each letter that we read, we plot a new dot in the middle of the segment determined by the current dot and the corner labeled by the DNA letter that is being read. So let me give an example, because it's a lot to keep in mind. Let's assume that we want to plot this sequence. A, C, G, C, T, G. We start with a square labeled with A, C, G, T. So the letters label the corners of the square. The first pixel of our figure is the center of the square. Now the first letter is A. So we're going to plot a new dot in the middle of the segment that is determined by the center of the square and the corner labeled A. So the first dot is going to be this red dot here. The second letter is C. So I'm going to look at the current dot and the corner label C, and I'm going to draw a red dot there. The next letter is G, so it's going to be somewhere there. The next one is C, it's going to be somewhere here. The next one is T, it's going to throw us somewhere here. Next one is G, it's going to throw us here, and so on. So if you ignore the dotted lines, every DNA sequence is going to correspond to some dots in the unit square. It's going to correspond to some image, if those are pixels. Okay, so let me give you some examples of this. So first of all, what is the correspondence between the CGR and the original sequence? So there is a theorem like early on that proved that it's not a difficult proof either. Given an exact coordinate of a CGR point in the plane, we can determine the letter that it came from, right? You can go to the last. So if you have the last letter of the sequence, you can basically, the last dot, the dot corresponding to the last letter of the sequence, you can basically determine the entire sequence in principle, if you had infinite resolution. However, in practice, computers cannot store exact coordinates of all CGR points. So in practice, we are always limited at some resolution, and some dots fall in the same little square. So it's not exact. You cannot really retrieve it. This is like a theoretical result. All right. So how do we store this matrix, this picture? Well, we can store it as a matrix. So, if you think of the square, this corner was labeled A, this was C, this was G, this was T. So all the A's are going to fall into this corner, all the C's are going to fall into this corner by the geometry of the chaos game. So, and you can store that as a matrix. For example, a frequency CGR of order 2 is going to be a matrix, 2 to the 2 times 2 to the matrix, in general 2 to the n by 2 to the n, or 2 to the k by 2 to the k, where, in the small little corner up there are going to be all the CCs, in the next one are going to be all the GCs, and so on. So basically, a CGR representation is a simultaneous representation of the k-mer counts. So in the end, depending on your sequence, how many CCs you have in them, all of them are going to be counted here, all the TTs are going to be counted there, and so on. So it's like a simultaneous representation of those, but of course they're also, related to each other. So if you want to normalize, because you want to plot different sequences, different length sequences, you can normalize by length so that you can compare short sequences with long sequences. Okay, let me give you some examples. So these are some CGRs of computer-generated sequences. So these are, if I remember correctly, all of them are representations of sequences of length 150,000. So the first one is the representation of a random sequence of length 150,000. So this is actually very good because a sequence that is random looks random. It should look random if you want to help our intuition when we look at these pictures. And this is not true for all the polygons. For example, if you play chi-square representation on a triangle and you plot 3-letter sequences over a triangle, actually a random sequence is going to look like a Sierpinski fractal triangle because there are some places in the triangle where you can never get if you just plot the middle. Okay, so that's not good because it's misleading. So the good sort of coincidence here is that a random DNA sequence will look random. This is A to the n, where n is 150,000. And as you see, you have like lots of dots, bunched on top of each other. These are 150,000, right? But all of them are going to be like all of them bunched on top of each other there. We have limited resolution, as I said. These ones are ACGT to the power n over 4. So you see, you have some of them there. All the A's are going, all the G's are going to be there. So they're going to be grouped like that. These are, I think that this was a sequence that is missing all A's. So if it's missing all A's, nothing is going to fall in this part of the sequence because this was the corner labeled A. And similarly, in a flactor fashion, nothing is going to fall there. This is a sequence missing, I think, A's and C's. This is missing T's and C's, and so on. So you see, you can get some insight into the sequence just by looking at its visualization with CGR. Well, so these are, if you are to be working in theoretical computer science, like I did in my previous life, there are all kinds of sequences and words that people spend their life studying, like Fibonacci words, Bonacci words, 2 Morse words, Lindon words, De Bruijn words. So we were curious to see what these mathematical sequences look like. And in my view, they all look very boring. If you look at this one, I think this was the Fibonacci word, Fibonacci word. You spend all your career studying that. Look how periodic it is. Like, who cares? No, this is what I'd say now, not before. So you see that all the mathematical sequences that people study are either very periodic, so they don't have like complexity from the point of view of CGR, or they are almost random, which are also not interesting. There is nothing sort of in between. If you look at DNA CGRs of biological sequence, the picture is entirely different. So let me show you what real DNA of real CGRs of real DNA look like. More interesting, right? So this is the CGR of a nuclear genome for Homo sapiens. And this is the CGR from archaea, the second kingdom of life. You have bacteria, archaea, and eukaryotes. This is one from Archaeoglobus fulgidus. So you see they have interesting patterns. What do they mean? Are they similar to each other? If you take things from the same domain, do they look similar or different, right? So there are all kinds of interesting questions arising. So actually, I wanted to show you just for fun, because there are 6 kingdoms of life. Let me give you a representative of each. So this is A is human from kingdom Animalia. This is, I think that this was plants, and this is protists, and this is bacteria, and this is archaea. No, I think this was plants. I don't see the caption there. So, this is one from each kingdom of life. You see, they look very different from each other. So, and remember that this is not taken from a gene or from any particular point. This is sort of similar along the genome. And actually, this brings me to my point. So the interesting thing about this chaos game representation of DNA sequences is they can act as genomic signature. In what sense? In the following sense. Real life genomic CGRs are species specific, in the sense that the patterns are similar for DNA sequences taken from the same genome. So even, the human genome is like a 3 billion base pairs, right? It doesn't matter where you take your sequence. It's a different sequence. But if you plot it, it's a similar pattern, right? Along the genome, the picture looks the same. And this happens for everybody, not only for human, right? For any organism. And moreover, the patterns are different, sometimes visually different, if you take DNA sequences from different species. So these patterns are also preserved regardless of the length and location of the sequence. So this means that if you have a very short sequence, you are going to have a very faint pattern. If you have a long one, it's going to be much darker, but the patterns are going to be the same. So it's a little bit like a watermark that can characterize the genome. So these properties qualify this CGR, chaos game representation of a genome. It qualifies it as a genomic signature, right? So we can use that, therefore, to classify, new genomes and, try to figure out what they are, if they are novel ones. So our first method for species classification, just sort of crude method, was the following. So we start with a bunch of DNA sequences. Here there are about 4,500 DNA sequences, and we compute the pictures for all of them. We compute the CGR for all of them. Secondly, because we didn't know better at the time, but it ended up working pretty well, we figure out, okay, these are images. Let's use distances from computer vision. So there are lots of distances available in computer vision. We picked one, structural similarity index, and we computed the distances between the CGR as if they are photographs, as if they are pictures. This gave us a pairwise distance matrix. So we have a distance between anybody, everybody, and everybody. And then we can use a method from statistics called multidimensional scale that does the following thing. If you give it a big matrix of all pairwise distances between a set of items, it outputs like a 2D representation where every dot corresponds to an item, and the Euclidean geometric distance between any two dots is roughly similar to the distance in the distance matrix. It's not the same because the original space may not be Euclidean, but at least it tries its best to make them, what is short should be short, what is big should be big, stuff like that. And actually, we did sort of better than that. My student Ralis is better than that. He had a 3D representation where I think I have it here already. Okay, so this is a representation, molecular distance map, we called it, of vertebrata, where dark blue are fish. This teal is amphibians, green is reptiles, red is mammals, and yellow are birds. So you see, you can rotate and you see what's going on. You can zoom in, you can zoom out, It's a little bit like Star Wars here, you can have it come at you. You can also search. So actually we are quite interested into seeing what happens. So first of all, we are very happy when we got this, okay? Just think about it. You know nothing about this organism whatsoever. All we have is in this particular case, we had a sequence that was 16,000 letters long. 16,000 compared to the billion base pairs of the genome is nothing. So all you know is one sequence of 16,000 letters, and you have about like more than 4,000 of these sequences. And look at it, it's like the classification is almost perfect. So this genomic signature counts something, captures something that is real and that can differentiate sequences and organisms between each other. We are also looking to see what's at the intersection. So if I try to see here Polypterus, okay, so this is a very cool fish that has lungs, and if there is a drought, then it can walk between lakes. It lives in Tanzania, and if it's a drought, then it will use its little fin and walks between lakes to get to some water. So it has some lungs, primitive lungs that can function even though it's a fish. So look at this one. It's a fish, but it's so close to amphibians, okay, that do breathe, that do have lungs. So these things that look like errors, maybe methodological errors, some of them are not methodological errors. Some of them are because, you see it is this magenta one here. It's like right there in the middle of a fish and amphibians. Like, why isn't it clean? Why is it one and another? Well, because it has characteristics of both. So this method really captures something. Okay. So that was what I wanted to tell you about the mathematical representation. Now, how can you use this? So, we were happy with that visual representation, but we couldn't really quantify it well. I mean, we could tell that some things are separated if you could draw a plane between them. But other than that, we had no way of measuring accuracy. Plus, some of this, we thought, the real space is n-1 dimensional if you have n items. So if you flatten it onto 3D, some of them are not real errors, right? Some of them would be perfectly separated in the space, but they look that mixed up in this one. So enter machine learning. So instead of just visualizing, we started to use machine learning, supervised machine learning, and then unsupervised clustering for taxonomic classification. So let me tell you first about the supervised machine learning part. So we all know how supervised machine learning works. In our particular case, we train the algorithm on a training set of pairs, and for each pair, we have a DNA sequence or its representation and the species name. This sequence comes from a cow, this sequence comes from a bird, this sequence comes from a reptile, and so on. And we test the power to predict the species of unknown DNA sequence after it was trained like that. Now, I just want to make a point here, because this makes this method completely different from alignment-based methods. So when I talk about the DNA sequence, what do I mean by that? In general, we take a random fragment from the genome. Okay, it has to be at certain length. So usually it works better if it's like at least 2,000, then you can see a pattern with the naked eye. But definitely it doesn't need to be the whole genome. Doesn't need to be a gene, non-gene, coding, non-coding, junk, it doesn't matter. So this DNA sequence, you have to extract a random fragment from the genome. Okay, so let's see how this worked. So this is our algorithm. So first of all, we converted each DNA sequence into a numerical vector. I'm gonna mention that. So for this particular paper, we used the numerical vector where every letter was assigned a different number. We didn't use CGR. We also have a version with CGR, but for this one, we used the other representation. Then we compute the discrete Fourier transform of the discrete digital signals. We compute pairwise distances between the magnitude spectra of DFTs. And then we use the distance between each sequence and all the other sequences as its feature vector. And, to train a supervised machine learning classifier. So let's see what we got. Well, so first of all, in the literature, there are many numerical representation proposed, okay? Like, for example, the first one called integer assigns T0, C1, A2, and G3, like a random assignment of numbers to the letters. Some other people went like, really, specific. For example, this representation gives some numbers that are equal to the atomic number of, of the nucleotide, whatever that means. Some others look at the electron ion interaction potential and give them some numbers related to that. So, we have like all gamma, either something that is meaningless or something that, tries to capture some biological significance. So there are like 13 different representations. Some of them are very odd, but very interesting. So one representation, just A, gives the letter A1 and all the other letters 0. So it only kept the positions of the A's somehow. So we tried all of them, okay? And as an example, this is the discrete digital signal of Canada goose versus European beaver. So the beaver is red, the Canada goose is blue, of 100 base pairs in the mitochondrial genome. So this was the 1 minus 1, the primitive representation, where 2 of the letter are assigned 1 and 2 are assigned minus 1. So you see, the graph is either there or here. And this is the magnitude spectrum. And then there are methods to compute distances between the spectra, and this becomes the pairwise distance. Okay, so this is what we got. We had like lots of data sets, of all kinds: primates, fungi, plants, amphibians, insects, vertebrates, and so on. We tried 6 different classifiers, and we tried all the mathematical representation, and we did like average over everything. So the best classifier for these data sets was quadratic SVM, and to our sort of surprise, the best mathematical representation was where T and C were given value 1 and A and G were given value minus 1. This is not intuitive, because you'd figure that you lose some information, but this one had the best. So what was it? The average was 90.7 accuracy of that. What was more interesting to us, and we still don't know the answer to that, is that one other, the just A, where A gets value 0 and everybody else value 1 and everybody else is 0, did almost as well. Sorry, this was the best one, was 92.3, 91.9. This is where you only see where the A's are. I don't know why, and the biologists don't know why. The only thing is that maybe because this is, you still remember some positional values, where everything is, you don't just count. Maybe the position where A is matters more than everything else. I don't know. Maybe for other data sets, it doesn't work. But for this one, we still don't know why this representation that loses so much information works, but it works. Okay, so this is just some accuracy and time comparison with other alignment-free taxonomic classification algorithms. I'm not going to go through that. The accuracy is better and the time is way better than other methods. Some of them we have to stop because they were like more than 14 hours, so we just have to stop them. So the method does really well. The accuracy is very good in the mid-90s, and then it's just like it works in minutes. All right, so as a corollary of that, in January 2020, there were like a few sequences of COVID-19 genome available. So I had one enterprising PhD student, Gurjit Randhawa, who thought, why don't you try to classify that? Okay, so here it is. The genome of COVID-19 is 30,000 letters long, and it looks like that. So this is the CGR if it is that. So this is the enemy number 1. Okay, that's what it looks like on a wanted poster. So what we did, okay, so it's in general, it's a difficult task to have a big data set of all kinds of things and classify directly at the species level, because you have too many clusters, right? So you can have errors because, each species, we are going to have just like a few sequences of it, right? And you have thousands of clusters. So instead of doing everything directly at the species level, we did a hierarchical classification, where first we took all viruses and we did it in the virus families, and we figured out, aha, COVID-19 is riboviria. Let's kick out everything out and zoom into riboviria. And then you classify riboviria into its subclasses, whatever there is, and aha, it belongs to coronaviridae, and so on, until you figure out that it's part of beta coronavirus, and is the sarbecovirus and part of beta coronavirus. So the accuracy were pretty good. These are like at each level of hierarchical classification, 95, 93, down to whatever. At this point, it was 100%. And, the interpretation of that is that, this was a comprehensive in minutes accurate analysis of a data set of almost 6,000 unique viral sequences, including 29 COVID-19 virus sequences, which was the only thing that was available as of January 2020. And our results support a hypothesis of bat origin and classify the COVID-19 viruses line with sarbecovirus within the genus beta coronavirus. So this was the effectiveness of this method applied to a specific problem, the COVID-19 classification. Okay, now, of course, there are downsides, and we all know what are the problems with supervised machine learning, right? You know, the fact that, you know, it can only recognize something that it has learned. So if it has seen something that it hasn't learned before, it's going to predictably classify it incorrectly. And this is a specifically acute problem for biodiversity, because remember, we only know 5%. So the chances that you get something that you've seen before are not that high in general. So we developed other unsupervised learning methods in order to sort of cluster things, sequences that we don't know anything about. So this is what I wanted to talk about now, unsupervised clustering for taxonomic identification. This is Deluxe, which is the work of two of my students, Fatima Alipur and Pablo Mijana Arias. And this is what we did. So for this one, so this Deluxe, it's a good name, right? Deluxe. Right? So you try to, you know, cherry pick our letters to make something nice. It stands for Deep Learning for Unsupervised Clustering of DNA Sequences. So the mathematical representation that we used was a frequency K-game representation, that is the K-mer counts. And what we did, because of course you have no labels for clustering, you have no labels for any sequences. So how do you learn anything? So we tried to, we produced, you know, we use data augmentation. That is, for every original sequence, we generate what we call mimic sequences. So a mimic sequence is formed from the original sequence by making some mutations in it. Right, so we had a certain percentage of mutation, and this mimic sequence and the original one was deemed as a pair and deemed to belong to the same cluster. So we know nothing about nothing, but we know that the original and these mimics that we just introduce a bit of noise, those have to belong to the same cluster. And then, it was like a loss function that had to be minimized. And at the end, there was a big variability in the results. So we used the majority voting scheme to determine this. So these are the papers from 2022 and 2023 that appeared about this. Okay. So conceptually, where does this stand? So the previous method, machine learning with digital signal processing, was supervised learning. So in the machine learning based alignment free genomic classification method, it sits here in supervised learning, and then Deluxe is, it's unsupervised deep learning or unsupervised clustering. Okay, so that is the outline of the method. So we have the sequences. For each sequence, we generate mimics. We introduce some mutations. Then we compute CGR representations for all of them, for both the originals and the mutations. And we feed the algorithm pairs of original and mimic sequence to several A and N's. And then, with their loss functions. And at the end, whatever prediction we got, we use for cluster assignment, we use the majority voting to decide the cluster assignment for each sequence. So what did we get? So this is like just an example of the data sets that we use. For example, if you have the same data set of vertebrate mitochondrial genomes, here is the learning process for the clustering of 2,500 of them into 5 clusters. So here are the epochs. The position of a point indicates the probability that it is assigned to different clusters. So each corner is a cluster. So we have animals, amphibians, reptiles, birds, and fish. So we have here a pentagon. So in the beginning, everything belongs to every corner with equal probability. And slowly, by epoch 149, all of them are perfectly assigned to their clusters. These are some comparison, the comparison with other unsupervised clustering methods and also with supervised method. And it did pretty good. It sort of did better than the unsupervised ones. For some data sets, it even outdid the supervised classification, which is a little surprising, given that here you have no knowledge about any DNA sequence. So this was this unsupervised clustering, which is very useful when you don't know much about the data set or about the data set is sparse. Okay, so these two approaches, supervised classification and unsupervised clustering, in these papers and many other papers by us and by others, give a definitive answer to the first question that I wanted to ask. So the question was, to remind you, Does biological relatedness produce a detectable mathematical signature in genomes? The answer is resounding yes.
I'm gonna start tracking.
So there is a
Do you need the flow hood for something before?
It's like a watermark. Okay. So it doesn't matter where in the genome, there is something that can be detecting and can be used to classify. So now, how do I think about it?
Yeah, because if you do it today, tomorrow won't be.
I think somebody has to put their microphone.
Yeah, maybe tomorrow, so they grow over weekend.
The way this works is as follows. So think of that you have two books in English and French, and you know neither English nor French. So you don't have to think about what they mean, what they are about. Even if you don't know anything, if you take an arbitrary page from one book, you know, you rip a page from one book, you rip a page from the other, and let's say you count all the words of length 3. Like the English is going to have lots of the, right? And the French is going to have le. And based on that, you can tell, okay, they are not the same. This is English, this is French. I think something similar is at work for DNA, because DNA is a language after all. So it has, even though you don't think about genes and what they do and their function, there is still some sort of a syntactic measure, the word usage, that can allows you to distinguish, you know, if they are distantly related, they're like languages that are distantly related, or you know, they are maybe like dialects of each other, if they are, you know, subspecies, but you can still tell them apart based on that without knowing anything about the content. Okay, now I want to come to the, you know, what is for us the more recent and most exciting part of this talk. We started to ask a different question. So all the papers, all the research that has been done so far into the genomic signature could attribute, you know, the assumption was that this difference is attributable to biological relatedness. So why is your genomic signature similar to that of a whale? Because you are both mammals and you have a common ancestor, right? So it's based on common ancestry, it's based on evolution, tree of life, and so on. So it was assumed that 100% of the signal is taxonomic. It's based on ancestry, common ancestry, and relatedness. But, you know, we thought, is there something else? Maybe you can pick up a signature based on something else. For example, could the environment produce a detectable relationship that is independent of biological relatedness? Could the environment produce a detectable mathematical signature in genomes? So we started to ask this question. I think the answer of this question is no, if you talk about normal environments. But then we had this idea, how about we look at extremophiles that live, maybe if the environmental signal is very, very strong, maybe then you can detect a signature in the genomes. So we started to look at the extremophiles. So what are extremophiles? Extremophiles are organisms that are able to live and even thrive in extreme environments where most of everybody else would be dead. For example, extreme temperature of over 100 degrees Celsius, or radiation, you know, or in something that is extraordinarily salty that, you know, you wouldn't be able to drink it, or very acid. And there are lots of these organisms. For example, you know, some of them live in hyperacidic lakes and volcanoes, or in hot springs, or in Antarctica and ice. So there are lots of extremophiles out there that live where everybody else would be dead long time ago. So we looked only at 2 kinds of extremophiles data sets, namely the ones that live in extreme temperature and extreme pH, that is extremely salty or extremely acidic, alkaliphiles or acidophiles. So these were bacteria and archaea. This is the, you know, the summary of the data set. And how do we extract the signature of this genomic signature? For each of them, we took an arbitrary 500,000 letters long genome fragment. So the genomes are very long, they're millions. So we picked 500,000, you know, random fragment in the genome, and then we took that to be as representative. And so what do we have? We have like archaea and bacteria. Remember the two out of 3 domains of life? And psychrophiles are the guys who live in ice, who like cold, mesophiles, so-so, thermophiles, they like it really warm. Hyperthermophiles like it really hot. And bacteria the same. And then for both of them, for bacteria and archaea, we have a data set of acidophiles. They like really high acidity, or alkaliphiles. They like the opposite of that. And we know we try to answer this question. So let me remind you just once again, because we didn't, so this study does not look at eukaryotes, But I just wanted to remind you how vastly different taxonomically they are from each other. So bacterium is more different from an archaean than a polar bear is from a flower, okay? They are more distant from each other. They're really, really, really different taxonomically, right? So this is crucial to interpret our results. So just to, to look at the terminology. So for psychrophiles were the ones for which the optimal growth temperature was under 20, mesophile between 20 and 45, thermophiles 45 to 80, hyperthermophile over 80 degrees. Let's remind ourselves that we are 36.5, so we ourselves will die here. Acidophiles, the neutral pH is 7. Acidophiles have optimal growth pH less than 5. Alkaliphiles, they have a high one greater than 9. So this is just to clarify terminology. And let me give you some examples of these folks. So this is my very favorite. This is Pyrococcus furiosus. Now, this is an archaeon isolated from heated sediments on Volcano Island. So it was near a volcano in Italy. It's a hyperthermophile. Its optimal growth temperature is 100 degrees Celsius. And it's called Pyrococcus because it looks like a fireball like that. It's like brightly colored. And furiosus, it swims furiously at over 100 degrees. So it's like has like this rapid movement. So this is like an example of extremophile. Let me give you some others. So actually, this was nice. So there was an article in Western News about this research because my collaborator is at Western, and it was published in Western News on Halloween, and they used this origin black. So it's like a very serendipitous choice. Let me give an example of another one. This is Psychromonas ingrami. It's a bacterium isolated from Arctic polar sea ice. So this can live at like below freezing temperatures. So this is a psychrophile. Its optimal growth temperature is minus 12 degrees Celsius in salty water, obviously, because otherwise it would be solid. And this is the lowest recorded growth temperature for any organism. So the name of psychrophiles, and this one comes from the Greek psychros, which means cold or frozen. This is the one that likes acid. This is Picrophilus torridus. I got to love this name. It's really torrid there. So this is actually a polyextremophile. So this is an archaeon isolated in a hot spring in Hokkaido. This is an acidophile. So this pH is very acidic. Remember that the neutral is 7. So this is 0.5 is like really very acid there. So its name comes from Greek pikro, which means bitter, and torridus, which means hot. So optimal growth temperature is 60 degrees. Some of these are polyextremophiles. It's both high acid and high temperature. Or some of them are high acid, high radioactivity, high temperature, high pressure, high everything, right? You basically cannot kill those things. These are very interesting because now that people are thinking of sending shuttles to Mars, so this would be candidates of organisms to send to Mars to survive outside of a spaceship. So there is like a lot of research about extremophiles lately because of this Mars space research. Okay, I think this is my last example, Natromonas pharaonis. It's an archaeon found in a salt lake in Wadi Natron in Egypt. So this is an alkalifile or halophile, thrives in extremely high salt concentration, pH of 11. So to explain this in ways that we can understand, this is a 300 grams of salt per liter of water. That's very salty. It's more than a cup of salt in a liter. Okay, and the thing loves that stuff, right? That's where they like to live. So this is similar to lye soap or the salt content of Dead Sea, which is called Dead Sea because everything else is dead there. Okay, so these are the extremophiles that we are talking about, and we have a data set of about 700 of them. So these are some of our results. So these are the results of the supervised learning classification of the temperature extremophile data set. So I have here various values of K. So we looked at subwords of length 1, 2, 3, 4. So we started to see a signal, the best signal is around 6, but you can see the signal even earlier. So let me point you to our results. I'm not going to look at all the classification models. I'm just going to look at the first one here. So when we train the supervised machine learning with taxonomic labels, bacteria or archaea, the accuracy of the classification was splendid. Okay? It was 99.5. Predictably, right? They are so different from each other that you don't need much to tell them apart if you are a supervised machine learning algorithm, right? But to our surprise, if instead we took the exact same data set, we removed the taxonomic labels, and instead we put very hot, hot, middle, and very cold, we got an accuracy of almost 84%. Okay, there is a signal there. Especially since if you give random labels, you get 27%. So it's definitely not an artifact, because you say, maybe there's something wrong with our method. Let's assign the labels randomly, see what we get. If it's still high, there's something wrong with us. But it's not. The signal is still there. And it's very strong, it's like middle 80s. Okay, this is the supervised learning classification of the pH extremophile dataset. Again, let's look at the k equals to 6. The accuracy of classification by taxonomy, again, very high, expectedly. By environment, if you strip the taxonomic labels and the very same data set, you give it environment labels, acid or salt, it's 92%. That's very high. This means that, again, that there is some sort of a word usage, some pattern that distinguishes, high salt from high acid, which is very unexpected, all right? So, these are some, there are some interpretability methods that are attached to some of these supervised machine learning algorithms. So this is a depiction of the global importance of various sigma in a one versus all classification by environment category. So if you want to classify hyperthermophile versus everything else, what are the sigmas that are the most important? So there are these ones, in dark blue here. So you see, for a different classification, sorry, this was hyperthermophiles. These k-mers are important, maybe overrepresented, maybe underrepresented, that distinguish hyperthermophiles from everybody else. Note that the thing that distinguishes psychrophiles from everybody else is different. So each of them has a different signature. And the same for acid, right? There are different k-mers. These are like not that many, but if you look at the quantitative ones, you can get a list of k-mers and you see that they are really different. These are, some histograms. You wanted to look at the deviation this time of k-mer counts from the mean. So, if you look at the signatures of psychrophiles, mesophiles, thermophiles, hyperthermophiles, and you look at the deviation of the 3 metals from the mean, you see that they are different. For example, the psychrophiles, you see this one, whatever it is, AAA is overrepresented here compared to the mean, while in hyperthermophiles is very underrepresented. So each of them has a different k-mer's frequency profile, different k-mer profile. And the same for, you see the different signatures also for the pH data set. Okay, so then let me tell you what we did afterwards. So, this hypothesis was so unlikely that my main role in all this was to say, I don't believe it. Okay, I don't believe this. So you have to ask, there is something wrong with this. It cannot be. There is something wrong. Try this, try that, try that, try that. So we literally tried everything. So Pablo, who was the lead author on this, was at the receiving end of all my, doubting Thomas questions. So we really didn't believe that. So what we did, we said, maybe we are doing something, overfitting. I don't know what it is. Maybe we are doing something wrong. Let's try unsupervised clustering. So let's throw, for example, for temperature, let's throw all the bacteria and archaea and don't tell, no labels at all, no taxonomy, no environment. Let's see, what does the algorithm thinks based on what cluster does it form, based on knowing nothing about nothing. So to our surprise, we absolutely expected the clustering to be based on bacteria or archaea, or at least bacteria hot and cold, archaea hot and cold, or something like that. We didn't get that. We got some clusters that are stable by multiple methods that contain bacteria and archaea together as being similar in their genomic signature. I don't know, like I'm going to say this again, this is like so surprising. So this shouldn't be similar at all because they are so distant from each other, but they are clustered together by this algorithm, by clustering algorithm. So this is like saying that, if you are extremophile, your DNA live in the Arctic, let's say, then your DNA is more similar to the DNA of algae that lives in the Arctic than to the DNA of your cousin. You see, it's like absurd, right? But for extreme environments, it does happen. So we did like a multiple different, challenging scenario, and this came again and again and again, and I wanted to give you some example. This is an example of the things that were deemed similar, even though they are completely different. So we found a hyperthermophile bacterium clustered with archaea no matter what we did, no matter how we tried to poke holes into it. So one of them is Thermocrinus ruber, which was isolated in Yellowstone in aquatic thermal hot springs. You know, they are very nice and colorful because they're so acid, nothing lives in there. So they have these beautiful colors. So this Thermocrinus rubber, there are these pink filaments around here. There's a pink color in Octopus Spring in Yellowstone, and 3 archaea. 2 Pyrococcus, this is the same Pyrococcus furiosus and another related one, and another one, Thermococcus littoralis, which was also isolated from thermal hot springs. So you see, in principle, the green guys and the yellow guys should have nothing to do with each other. And why were they grouped together? Well, let's look where they live. They all live in very hot places. Either, you know, in a hot spring, near a volcano, or in ocean, in a hydrothermal spring where like hot water from the inside of the earth, you know, comes to the surface, or again in a aquatic thermal hot spring. So there must be some similarity in the genomic signature that is due to the environment, and that trumps the biological relatedness. So these are just, you know, we try something else. I said, you know, maybe there is some mistake. Maybe there's some sequence similarity. So we tried to feed this into alignment-based tools, such as BLAST. Maybe we made the mistake. Maybe somebody entered the wrong sequence. You know, I was still trying something wrong, something wrong, something wrong. But we tried to do a query with the Thermocrinus ruber in bacteria and compared to the other archaea, 0 similarity, right? There is 0 hits. Okay, so they are not similar. Well, you know, based on genes, aligned genes, or anything like that. It's just like this word usage pattern that somehow is pervasive. So, so then we look, you know, well, how are they alike? Why are they grouped together? And here it is, you know, all of them are from, this is the, you know, JGI gold environment description. All of them, it's either aquatic thermal hot springs or hydrothermal vents. So this is the common thing that belongs to all of them. Okay, so then, what is my question to the second, or my answer to the second question? Can the, okay, let me put it here, extreme environment, because I think that for small variations, it won't hold. You know, the signal won't be so strong. Can the extreme environment produce a detectable mathematical signature in genome? Surprisingly, the answer is also yes. And this is the paper with lead author Pablo and some, you know, my former student Gurjit and some biology students of Dr. Hill, that appeared in Nature Scientific Reports last month. So this was very surprising that the reviewers couldn't poke holes in it either. So I'm almost convinced that it holds. And I wanted to end by listing my collaborators. This is my biology collaborator from Western, Professor Kathleen Hill. This is Gurjit, who is the lead author of the COVID-19 paper and the MLDSP paper, the supervised machine learning. This is Maximilian Soltisiak. He's currently at Harvard or Stanford? Harvard, thank you. And he was working on extremophiles. This is Rallis. He is the one who did the molecular distance maps. Pablo is here in the audience, Fatima is in the audience, and this is Joy Butler, who is also the lead biology author in extremophiles. So I wanted to thank you for attention, and I wanted to show you some machine learning rendition of a photo of mine that I got at the museum, the Picasso Museum in St. Petersburg, Florida. So the machine learning is good. I don't think I'm recognizable, but it's still interesting. Thank you.
Thank you. That was very interesting. We haven't had questions. So, anyone has questions?
So it's a fantastic talk. Now, the reason that we talked about the sequences,
Yeah,
and consuming images. How much smaller sequences is that still as relevant as it's in the longer sequences?
yeah.
Because you can see the pattern, like let's say something that's 100 meters right now, you still is relevant.
I think that visually you stop seeing a pattern at 2000. I think that that picture was like very long. So 2000, you know, it depends on your algorithm. I think that there are some methods, for example, but they are not based on CGR. Okay, I think if you want to use CGR, then you cannot go too short. But you can try some other methods and apply machine learning directly to the sequence. And in that case, there is this bioscan project that we are part of that, like Pablo, Niusha, and Moneri are part of with the University of Guelph. They use a sequence called the DNA barcode that is 653 letters long from a specific gene and a specific location. And they use that to identify organisms. But you see, for me as a mathematician, this is not satisfying because, this is not generalizable. So this works well for kingdom Animalia. But if you go to plants, they don't, that barcode doesn't work. If you go to bacteria, they don't have that region at all, right? So something that doesn't generalize and you cannot apply it to anything, for me, I mean, it's good in practice, okay? Like our colleagues and we love them, but you see, mathematically, for math, that's not satisfactory. While this method, you can throw anything. You can compare bacteria with cucumber, with human, with computer-generated sequence of your choice, with Fibonacci word, right? And you still get something. For me, that is very attractive, right? This like the universality of it all, that is applicable no matter what the scenario. Yeah.
I'm really puzzled. I'm Josh Rico, biology, biologist, and I sort of have a quick question about both halves of the talk. This here, the one with the visualization side. We were focusing on mitochondrial DNA and some comments about that as well. I was just wondering though, the resolution, what level of resolution do you actually have? Because it's a very different thing to be able to say that's a fish and that's a mammal versus that's which species of each of those you can distinguish down to. So I'm wondering what the relevance would be if you can only go to that high level. So
this resolution, so first of all, is the choice of K for K-mer, right? So we found that depending on the data set, we had success. I mean, if the data set contains groups that are very different from each other, you can get away with K equals to 3. But, for other data sets, usually I try K equals to 6. It works the best for most cases. Sometimes you have to go to K equals to 9 if the genome is very, very long. Then you have to go to 9 to get like a better accuracy. But at some point, if you take the K to be too long, then the accuracy drops because there are too many unique sequences. And then every sequence is, completely different from each other if you consider only. So all the sequences are maximally different if you consider only unique sequences, right? If you take K too long, you are only going to hit unique sequences. And then you're going to have distance 1. This one has it, this one doesn't have it, right? Distance 1 between all of them. And therefore, no classification at all. So it's like a little bit of a U shape, right? The accuracy increases until about 6, maybe 7, 8, 9, and then it starts to drop. Yeah.
About the environment testing. It's really puzzling. But one of the things that I wonder about is you think that maybe the gene C content of the genome would be higher for some extreme. Not thermophiles, though.
Right.
Not typically thermophiles.
Right.
Maybe something like that. Anyway.
We tried that. We have an answer because one of the reviewers was obviously a biologist. That's exactly that. So this GC content corresponds exactly to K equals to 1. Because we took, well, actually, when we did a study on extremophiles, we took both the sequence and its Watson-Crick complement because we were afraid that some people are going to upload the other strand. So GC content, G plus C content, right? Corresponds exactly to K equals to 1 for us. So for K equals to 1, the taxonomic signal was strong, but the environmental signal was not strong. Yeah. We got like almost like random, right? So for neither pH nor the temperature, we didn't get any environment signal at K equals to 1.
But I think it's the halophiles that have that GC content enrichment in the genome.
I'm not sure, Pablo, because I think that Pablo has like nice figures about that. We didn't notice. Because we computed the GC content specifically for each of the categories. I don't think I remember for.
There was temperature, we didn't get anything, and from pH, we didn't get any content, and from salt, didn't get anything. Like that's not included in the paper. Maybe we get it.
Just like halophiles, we have the halophiles, right?
Yeah.
We are the same as alkali files. So I think we got nothing. I'll be happy to send you the figures, okay? Because we computed the stats.
Yeah. Okay. Yeah,
but this doesn't say that some particular ones don't, right? It just said that like overall, we didn't see a difference.
Okay. And then the temperature one brings you closer to that last universal common ancestor, because that's what unites them potentially. That's where they originated from. So I wondered though whether that somehow is a reflection.
But that's taxonomy, right?
Yeah, that's right. Yeah, but then I may have to do the rates of evolution for different types of organisms, different types of environments that they have. But the percent.
I was having nightmare actually that this is not, I was just like, maybe the tree of life is wrong. Maybe bacteria equals to archaea, and we just discovered that the tree of life is wrong. But I don't think it's that wrong, right? I talked to some of my colleagues and it's pretty solid, right?
Yeah.
So if the tree of life is not wrong, then the environment signal exists for temperature. We tried many other things that I didn't talk about. We tried to train with thermophilic archaea with all the bacteria and all the non-thermophilic archaea. So then they were classified wrongly as bacteria, right? Because all the other archaea are not hyperthermophilic. So we tried many things, and if all the answers were consistent with that, yeah.
I just have a question on that same line of thinking. So you're talking about comparing images, creating algorithms and stuff, but for the environmental ones, did you take the sequences that you used to generate the images and do an alignment with just the sequences?
You couldn't do alignment because they're from different parts of the genome. You would get nothing.
But yeah, I guess.
So that there are probably coding regions because bacteria and archaea have upwards of 80% coding. So probably it's not non-coding. We didn't try. I doubt you'd get anything, right? Because they're.
So I guess along that same line, I know you're doing metagenomic assemblies and
Right.
from big environments. People will use things like epimers and different types of algorithms
Right.
Right. I was wondering if you thought about applying anything like that.
But we did that. We used all the k, including k equals to 4. So it's the same thing. By the way, we did the blast on it, right? So at least for those examples, blast gave nothing. There was no similarity in the sequence.
Yeah, I wouldn't expect it.
Right?
So it's smaller, but yeah.
Right. So that didn't work. If you can think of any other way to poke hole in this, I'd like to know. I tried everything I could think of. Because we didn't believe it. We worked on this for 4 years. We didn't believe it ourselves.
There's no mechanism. That's the frustrating thing.
Right. What's the why? That's the problem with the machine learning, right? but I have to tell you that we did obtain some corroborative evidence because for some of this we could extract the machine learning extracts what are the k-mers that contributed to that classification. And for many of those, I would say that for 75%, we found corresponding papers in the literature that for k equals to 3 that identify those codons as being overrepresented or underrepresented in some extremophiles. So we found for many of them, we did find for specific streamers, we did find corroborating biological literature.
So if all extremophiles share very specific genes.
Not genes. It's not genes. It's like word usage. It's like a streamers. It's codon usage there.
4 genes.
Yeah, maybe they would show up. Yeah.
Enough to train the genes. Well, that look for those.
We didn't try with the conserved genes.
Very.
I don't know if they're long enough for to have so that they show in en masse, but we could try the conserved gene. I think this is what we're monitoring. We have an extremophile 1' paper in the works, so maybe we are thinking of trying the 120 conserved genes for bacteria, right? Yeah, so this we want to try. Maybe.
Based on shared genes, the genome of those hyperthermophile bacteria. Yeah. So they have some core features that you find.
Yeah, that's interesting. So, but the thing that we were wondering about, but I think we're going to try anyway, that the conserved genes for bacteria and archaea are different. There are 120 for bacteria and about 53 for archaea. So it's not exactly an apple-to-apple comparison if you throw everything in the same data set. We will try. We can, of course, normalize by length. So we'll try that, see if we get something. Yeah.
Great. Let's let's in the form of the question and answer meeting. Of course, continue that, but let's thank you again.
Thank you.